Hello and welcome to the CAP Today commercial roundtable discussion for Wednesday, May 14th. I'm Bob McGonigal. I'm the publisher of CAP Today. I'll be your host for today's session. And I want you to know this is only our second ever installment of a new feature that we've thought up for CAP Today and for you as our audience and readers. This kind of feature is designed to present a company and a customer in tandem to discuss their work and solutions that they do. And it is sponsored today by Zeptometrix, and we're very pleased to be able to present this to you. Zeptometrix, as you know, is a very well-known company, and we're delighted that they've agreed to participate. Uh, and let me introduce now our two speakers. First, we have Dr. Jessica Van Allen. She's a PhD and a technical support lead for Zeptometrix, has lots and lots of experience in this area, and we're just delighted to have her on. And the customer speaker today is the very delightful Megan Starolis. Dr. Starolis is a PhD and is the senior science director for infectious disease at Quest Diagnostics. She's based in the Chantilly, Virginia lab, very well known. And I think you told me, Megan, you're responsible for about 14 laboratories in the Quest network.
It depends, but could be up to 26 at times.
Well, up to 26. So you almost never sleep, I assume.
Very little.
Very good. Well, welcome to you both. There's lots of things that we could discuss today, but one of the very key things I wanted to start with it was from Jessica Van Allen and talk about what are some of the really hot topics in molecular ID testing. And you mentioned in some of our initial discussions that viral load testing for transplant viruses is becoming ever more important in the field. Could you make a few remarks about that? And then I'll also ask Megan to comment for us.
Sure. Yeah. I mean, I think in terms of you can kind of approach it from two different ways, right? So there's kind of testing that goes on with LDTs or laboratory developed tests, or there's testing for FDA cleared assays. That's kind of the two big buckets of testing that goes on in with the labs that a lot of our customers are in. And recently, Megan and I both attended a conference, and one of the hot topics from a couple of the speakers was viral load testing with some common transplant viruses, things like EBV, CMV, VZV, some adenoviruses.
Mhm.
So that's always something, we've been playing in that field in terms of molecular controls for viruses for a really long time. So it's products that we've had on the shelf, but we're always looking to get that feedback from end users and customers in kind of how we can approach new products or kind of adjusting what we have on the shelf to support them in the testing that they need to do in terms of levels, strains, concentration, packaging that makes sense for a laboratory. So that's where those kind of meetings and getting feedback from labs like Megan's are really, really insightful for us.
Thank you. Thank you very much. Megan, tell me about testing in viral load for transplant related viruses within the Quest laboratories.
Yeah, I mean, one thing within Quest, but just with labs in general related to transplant viruses, is commutability of testing results. So
Okay.
with transplant recipients, oftentimes that patient is getting their transplant at a center of excellence or a transplant center that could be hours away from where they live. And one thing that we need to do with our patients after they've had their transplantation is we need to monitor them for latent viruses that may reemerge once they're on immunosuppressive drugs. So, um, up till recently, we've mainly had LDTs, lab developed tests for these types of viruses. And the lab could pick different calibrators, different stock material that could be quantified in various different ways, TCID 50, digital droplet PCR, copies per mil. And one thing we didn't have for a long period of time was an international standard. But we
Okay.
know that this is very important for transplant patients because as they leave the transplant center and they go back to their home, they might be getting testing done in various different labs.
Okay.
So having, standard tests that, available from the major vendors, but also having control material and calibrators that are calibrated in international standards will help us to be able to compare viral loads over time, even no matter what lab did that person's testing.
I see. But to go to Jessica back for a moment, it's going to be very important, though, that we have a well characterized control, is it not? And one that's
Right.
also, you've mentioned, I know, a number of times, the importance that we can have some quantitative results for the various controls so that we can do dilution studies and others. Could you just paint this part of the picture for me a little bit?
Sure, yeah, and especially when you're looking at things like viral load testing, you're looking for a specific quantity. And like Megan said, to that point of commutability, you want something as close to a patient sample when you're talking about a control or a calibrator as possible, so that you know the results you're being returned are accurate. So to be able to make clinicians are presented with this data and given these all this information to make these decisions on treatment and progress and stuff like that, it's really important that you have one, a control on the plate that you trust, that you know is giving you a correct positive or correct negative, and two, a calibrator on the same end that is giving you an accurate result and an accurate value. So it's really, really important. And I think we'll continue to hear about this as assays get more standardized and more FDA cleared tests, come on the market for these kinds of things, you'll kind of shorten that variability that can occur in between labs, which is great. But there's definitely what I gleaned from the talks that I've heard recently and anything I've heard in the last few years about this is that there's definitely progress that can still be made kind of to that end with these specific assays.
Very good. Thank you. Megan, you have stressed in our conversations the importance of really understanding what might be in a test kit or what even might be in an LDT quote unquote formula or recipe. Could you expound a little bit about that? We're talking, of course, when we talk to Zeptometrix, we're talking about a third party control with the ability to do lots of different things, as we've just heard, particularly with quantitative dilutions and ranges and so on. But talk a little more about the importance of understanding what a third party control is and isn't,
Mhm.
and talk about what's in the kit. That's one of your lines that I really enjoy quoting.
Yes. So I think it's really important that as lab directors, when we are choosing QC for our assays, that we understand the difference between an external control as defined by our regulatory organizations and third-party controls. So in my experience, having been a lab director, I feel like at times people use these terms interchangeably. So for instance, if there's an FDA cleared kit that comes with a control, a laboratory may think that they need to add another control as an external control that's a third party control. If you look at the definition from CAP of an external control, it is simply a control that is external to the reaction, as opposed to an internal control, which is part of the reaction. And in the CAP guidelines, it gives examples of what can be qualify as an external control. And it says it could be a kit control, it could be a third-party control, it could be a previously run patient sample, it could be a control that you create out of some kind of stock material. So as a lab director, when do you actually need to purchase a third-party control? I recommend doing a little bit of diligence with your FDA approved test at analyzing the controls that come with the kit. So in the IFUs, the instructions for use with the assays, it usually gives a pretty good description of the control material. It will say whole organism in this matrix, or it may say armored RNA in this matrix, or it could say nucleic acid. We want our controls, our external controls, to be full process controls. So we want them to control for the entirety of the process, including extraction, any pre-processing steps you do, or an amplification at the end. So if you have a kit control that is something like nucleic acid, that may not actually be a full process control if it doesn't require extraction. So when I'm looking at any kit, that comes with a control, I analyze what that control consists of. And if it is not a full process control, then I am going to select a third party control. And I'm going to reach out to someone like Jessica, and ask questions about that control. What is it made of? Is it a full process control? And does it actually control for what I think it's controlling for? Folks like her at these companies are really amazing resources to utilize.
Thank you. And Jessica, let me ask you to talk about your end of that very same question. Uh, it's easy to think someone just calls up the Zeptometrix 800 number and says, I need a control for yada, yada, yada. There's a little more to it, isn't there? And often the person on the other end of the phone perhaps has some questions they're too embarrassed to ask, but you'd encourage them to ask those, right? And you could give them a lot of help.
Yeah, definitely. And I think, it's not necessarily a direct email. So you're not talking to, we don't have to talk on the phone if you're not comfortable with it. There is an email that I get. I run the tech support email, and a lot of the questions come in that way. I do also interact with a lot of customers at conferences and meetings. But always, I give my card and say, please ask first, because there's so many instances in which someone just buys something because they heard from someone that this control they used one time and they just bought it thinking it'll work for them. And sometimes it will, and sometimes you're fine and no harm, no foul. But I think a lot of times, to your point, people are they don't want to ask a dumb question, which there's no such thing when you're spending money and trying to find something that's going to work for your laboratory and your processes. So I always encourage them. I mean, we try and put a lot of information on our website and our product inserts in the documentation that comes with our materials, but I always encourage people to reach out directly. Again, it might be a dumb question, but why would you take that risk of wasting money or wasting time in trying to get something going up in your lab when you don't have to? That's what we're here for. And that's kind of one of the benefits of working with a supplier of third-party external quality controls, is that we can do that legwork for you. We know what is in our controls, what their capabilities are. So why wouldn't you use that resource when it's presented to you?
Very good. That's an excellent point, and I hope we can underline that yet again. But Megan, I do want to talk to you first about a question that I'm sure comes up in your laboratory, because samples tend to arrive at the door, and it's left to you and your colleagues to figure out what it is that the samples are. What are the questions that the people have about the samples? And so let me choose one here. There are many different sample types that might arrive, some of which would be included in FDA approved kits as approved for, but then there's many beyond those. Could you talk a little bit about the importance of understanding the specimen that you're looking at and the appropriate use of controls and testing based on the variety of specimens
Mhm.
that you may see?
Yeah, that's a great question. So I will say that first of all, we do need to make sure that any specimen type that we accept is validated. If it's not part of the FDA approved kit, so for example, if the kit is FDA cleared for plasma, but we want to accept whole blood, the laboratory does need to do a validation of that sample type to ensure it works properly and yields expected results before we can accept it. But assuming that the lab has done that, and has their validation in place, um, I do think it's important to think about any controls you may need to add to your assay if the sample type that you've chosen requires any type of pre-processing. So an
Mhm.
example here that I like to give is for HPV testing. If you're using an assay that's validated for thin prep, but you want to be able to accept SurePath, which is another common collection media for Pap smears.
Hmm.
In many cases, there's a pre-processing step that's needed for SurePath. It could be like a proteinase K digestion that is different than what you have done with the FDA cleared samples. So again, going back to that requirement from CAP, your controls need to be full process controls. So if you have validated a sample type that requires some other type of pre-processing, you need to choose an external control that goes through the same process. Additionally, if you have validated a sample type that is very hard to extract, maybe it's something like whole blood. I'm giving that as an example, but actually
Okay.
our extractors are quite good at extracting whole blood these days. But if you
Okay.
have some kind of specimen type that's particularly hard to extract, then you may want to consider adding a control in that matrix to ensure that you have adequate extraction of your sample.
Good. Thank you very much. Now, Jessica, if a lab calls you and says we have a very exotic specimen, and yet it's the only one we have, uh, and ask, do you have some protocols or that you could share with our lab to help us adequately run this specimen using Zeptometrix controls?
Yeah, so a lot of times, if we have the organism in-house and we sell it commercially, I'm always happy to share what we do internally, the processes and the extraction kits and the instruments that we test on, with
Okay.
the understanding that, it might not be exactly what you do, but here's how we see it and how we handle it in our hands. And that is typically enough to get them started on the way. As someone who's worked in technical support for several years now, I do have a lot of feedback that I actually track and that I
Okay.
can go back to. So I'm like, oh, you know what? I did have a customer that was using this similar extraction kit. Here's what they did. Here's how, we troubleshot that. So I do have that kind of information. And like I said, I'll always share our internal information, whatever I can to help troubleshoot. But again, I do encourage people to reach out because one, it helps me gather information so that down
Okay.
the line, when someone comes to me with that same problem, I can be like, oh yeah, I dealt with that, and here's how we worked through it.
Very good. And along that same line, let me ask you, uh, someone might naively think that with a company like Zeptometrix, you just have a basic dinner menu. Here are the controls we have for these kinds of tests or these kinds of organisms, but there's a lot more to what you have on offer.
Yes, definitely. Like I said, we try to have a lot of information on our website as much as possible, but we do have a number of agnostic controls that should work across platforms and assays. We do have controls with known performance on specific FDA cleared assays. We have a bunch of live organisms and heat inactivated culture fluids and DNAs. So if it's not on our website, it might be an organism we got in a couple weeks ago for a custom growth of something that it turns out it's going to be something that's useful and it's emerging and we might want to turn into a product. Again, to that point of reaching out, not to like beat a dead horse with that, but
Hmm.
it's always worth asking that question. If we don't have it, can we source it? Or do you have a source for it and we can get it in and kind of work with it in our workflow and turn it into something usable for you? Those are all options that are on the table for us.
Very good. And Megan, tell me, at Quest, obviously, you've told me you have as many as 24 labs at some times that you need to be servicing for
Yeah, so we
infectious
actually
disease.
have 26 regional labs that
26.
perform
Okay.
molecular tests. We have a handful of our labs that are referred to as our esoteric labs, where we do our highly complex laboratory developed tests.
Okay.
But with FDA cleared test, we're doing that at, depending on the test, we're doing that at up to 26 sites.
Okay. So we mentioned briefly, but I think it's worth dwelling on. In many cases, you need to have some controls with organisms present at a very high titer so that
Mhm.
you can do dilution studies, correct? Which
That's
helps,
correct.
does a lot of things, including linearity. Uh, but tell me a little bit more about that. You're the expert, but I know our audience will be interested in what you have to say.
Yeah, we really like high titer stocks when we're doing both validations of LDTs and verifications of FDA-approved tests. So the amount that you have to do, the types of studies you have to do versus an LDT and a verification are different. But in all cases, we want to be able to have high titer stocks that we can do dilutions to test the linear range, so that in our accuracy studies, we're supposed to have high, medium, and low samples. We like to be able to, for our low samples, we need to get that quantitative value close to the sensitivity limits of the assay.
Mhm.
So having a material that can be diluted from a high titer, we can more closely control that. So we use these types of things for pretty much every type of validation or verification study that we do. And what I like to do at Quest is I like to work with a company to have a standard lot that has a known quantitation for all of my various sites so that I can help to oversee those validations or verifications at all of the laboratories that I support.
Okay, very good. And Jessica, I'm going to assume that as you mentioned, you can provide some of those high titers, but sometimes you're doing it with live organisms, sometimes attenuated organisms. Tell me a little bit more about this element of these important controls and the
Sure.
biosafety elements of them, because this is a considerable interest.
Yeah, definitely. I think a lot of times when people think of us, they automatically think of our ready-to-use QC material that's inactivated, kind of diluted down already in the matrix, ready to go on an instrument, which is a lot of what we do. But to Megan's point, we do also have a catalog of quantitative, kind of high concentration material, like she's talking about, where you can take it and you'll dilute it down into your matrix of preference to the level that you require for the work that you're doing. And when we don't have it, chemically inactivated in our natural format, we might have it as a live titered material or a heat inactivated culture fluid, something like that, that you can, again, in those instances and values, with a bacteria, it'll be in something like a CFU per mil value. With a virus, typically a TCID 50 per mil if it's a titerable virus. Whereas with our natural inactivated products that we're testing by PCR, you'll get a copies per mil or IU per mil value where possible.
Okay, very good. Thank you. Now we just have a few minutes left, and I know that Megan has a very important appointment right at the bottom of the hour. So I want to just turn slightly to discuss the other element of testing generally. I mean, we know we are always talking about a medical and scientific result, which is enormously important. At the same time, though, we're all operating with expenses, with income and expenses, hopefully a little more income than expense for everyone. So let me turn this and talk a little bit about uh, the cost of quality, or maybe another way of putting it is, what is the cost of poor quality? What do you observe at Zeptometrix? Do you see sometimes people have been trying to cut some corners and then realize they need to go back to some better practices, even if somewhat more expensive?
Yeah, definitely. It's a common thing that comes up, typically it is just someone having to decrease the window, the time frame in which they're doing their testing. They're not doing frequent QC, they're doing monthly or every other month, or God forbid, something like every 6 months and only using like a proficiency as their only QC.
Mm-hmm.
And you have to think about of course, the risk goes up depending on how many patient samples are being run, and how many results are giving back. If you're not running a ton, you're a small lab, it's probably not, that big of a risk. But if you're a lab that's seeing dozens or hundreds of patient samples a day, you have to really consider the impact of a false positive or a false negative result, that could have down the road, not only for the patient, but for the laboratory itself.
Very good. Megan, I'll let you finish your part of the session with us by talking about this very same question. I know that Quest has an enormous responsibility. The volumes that you perform every day and every night in the Quest labs around the entire country is staggering. So the responsibility then is equally staggering, I'm sure.
Mhm.
So talk about this cost of quality and what you're doing using controls to help give us the highest possible quality at the best possible cost?
It's a very important topic because something that we talk about very frequently at Quest is the cost of poor quality. Saving pennies and having a major issue can cause very, very big downstream repercussions. And this is something I've talked about in several presentations that I've done, is talking about, you know, it's not just the event. So if you have a major quality event that happens, it's not just the event itself. There are downstream repercussions. You could lose clients. You can lose trust from your physicians. Depending on the organism, you could have litigation that occurs. So the cost of poor quality can be really staggering. So what I like to do and what I encourage others to do is to really treat each assay individually. And when it comes to controls, don't be on autopilot. Really take some time to read your IFUs. If you're using an FDA approved test, take the time to really read it. When you're doing LDTs, and FDA cleared assays, take some time and do a risk assessment.
Okay.
If it's an open system test, it's more prone to contamination versus something that's maybe a closed system test. So don't be on autopilot. Really think about your specific test. What are the risks? What are the repercussions clinically of a false negative and a false positive? And then try to think about, do I need extra controls to control for these outcomes? But again, I really encourage reaching out to someone like Jessica because the control that you're choosing may not actually be controlling for the outcome you're trying to prevent if you do not have awareness of what's in that control. So if you're not sure, reach out to someone like Jessica who can, really advise you. Okay, you're worried about false positives. This would be a good control to use. You're worried about maybe a false negative because of genotype diversity of the organism. Maybe try this approach. So that we are keeping cost in mind. We're not over engineering our QC, but we're doing just the right amount to have impeccable quality.
Very good. I want to thank you very much, Megan. Dr. Starolis, I know you have a meeting to go to, so I'm not going to hold up the patient care that you're doling out at Quest every day, and I want to thank you for appearing with us today. I've got a couple more questions for Jessica Van Allen, and I'm getting them from some questions and answers from the audience. Let me ask you this, Jessica. What frequency would you recommend for testing using third-party controls in addition to manufacturer controls?
I think to a point that Megan just made, each laboratory has to take a look and do a risk assessment and kind of determine what the risk is. Obviously, if you're looking at an FDA clear test, you'll want to refer to the manufacturer's IFU to see if they give any guidance. A lot of times for them, it'll be something like when a new lot of cartridges or reagents is received in, you'll do your QC testing. But I think to Megan's point earlier, it does come down to doing a risk assessment in your laboratory and understanding kind of what a natural or useful cadence is for your particular laboratory and that particular assay.
Good, thank you. We have one question very specific for you, Jessica. Does Zeptometrix have any solution for HIV positive samples to run in a correlation study?
We definitely have HIV positive material, so please reach out.
Yes.
And I can definitely direct you to the right solution.
I thought that might be a fairly easy one.
Yes,
So
that was a
I'm
softball.
delighted. So as we approach the bottom of the hour, I just want to thank everyone who's been involved. I want to thank Zeptometrix for sponsoring this very great roundtable, for providing us a wonderful, wonderful speaker in Dr. Jessica Van Allen. Jessica, I hope we keep you very busy at the technical support desk because having seen this, if I had a question, I would want you and not so when I called in, You've done a wonderful job for us. And of course, I want to thank Dr. Megan Starolas. She's the Senior Science Director for Infectious Disease at Quest Diagnostics, and obviously a great expert and a real resource. And finally, in addition to thanking Zeptometrix and our two speakers, I want to thank everyone who's participated today. We really enjoy having you. We hope that this kind of new format and new offering will be of value to you. So we want to hear from you. Do let us know. And with that, I'm going to bring today's roundtable to a close. Thank you all very much.
Thank you.
you.